information to cover. So, as always, feel free to pause. Like, really, this is just for you guys, and everyone else gets to reap the secondary benefits. We have this outline that's provided on the website, sort of written down here. It goes through the different sections, and what I did basically was copy and paste this into a OneNote document where each OneNote page is a big section. And then as I went through, I just went to that subject and filled in details. So I kind of like reverse mapped what is the content that exists into the outline that they provided. I also looked at some like old practice tests that they had and similarly kind of mapped out the knowledge that was in this test into the sections here. And so what we will go over today is everything. And most of what you'll see is the stuff that existed in any of the existing question banks. If something didn't appear, I didn't consider it high yield. And so there may be some sections which are pretty empty, but that mostly meant that there wasn't much to know about it. Okay, so we'll just dive into it. So I'll zoom in a little bit here, but the first section is radiation interaction with matter. And it says definition of ionizing radiation-free radicals and radical damage. I think the thing for you guys to know is this minimal energy to ionize an atom, 10 eV, that'll be a flashcard. And then minimal energy to break an atomic bond. This happens with UV radiation. Then there's some, so the last thing I did about this is I listened to the Wallachek RadBio review. And so any information that she had in her presentation, I also tried to include in this. So you can consider this kind of like the Wallachek plus. So there's some information about radiation from natural environment. And in the US, it's higher in Colorado where there's mines and lower in some places like Florida, but the average is 3 millisieverts per year. And like, which place in the world has the worst radiation exposure? The worst is Ramsar, Iran, with average of 10 millisieverts and up to 260 millisieverts per year in certain areas. The highest natural source of ionizing radiation is radon gas. And then here's some other sources which contribute a small amount of radiation, including potassium from bananas. The biggest exposure by far is man-made, and that is medical. So 3 millisieverts per year due to CT scans, nuclear medicine, IR fluoroscopy, conventional radiography. And I think this is an order of biggest contributor to lowest contributor. And so you'll see that about half of the US exposure is just natural and then half is medical. And then for reference, one plane flight across the US is 0.35 millisieverts. Here's the summary, and that's that section. Next, we go into actually describing direct and indirect action of radiation. Some of this we actually covered pretty well, so some of this should be reviewed, but there's low LET radiation, like X-rays, and 2/3 of damage is indirect and 1/3 of damage is direct. Now, interestingly, like what's the distance that indirect action can happen? So you irradiate water molecules, you have secondary hydroxyl radicals. So it's 2 nanometers from the DNA. And then this causes base damage and strand breaks. And then on the other hand, there's high LET radiation like neutrons and protons. And these are almost all direct action, and almost all of it is alpha damage. Alpha damage is damage contributed from low doses in experiments. Linear, thought to be related to single electron causing multiple hits. Beta damage is damage contributed from high doses in experiments. It's quadratic and it's related to multiple different electrons causing synergistic hits. Throughout this time studying, you guys will wrap your head around alpha beta many times over. So there are different ways to think about it, but at least in terms of like alpha damage and beta damage, these are the definitions that I stuck with. You'll probably get like what phase is the cell most sensitive to radiation. So it's most sensitive during M and G2 phase. Rarely would you get something where it has like both of these because it's not like super clear which one's the winner. I would say M phase is the most sensitive. And then which one's the least sensitive, S and G1 phase, and like the least sensitive of them is S phase. During these different phases, there's different predisposition to kinds of damage. So as you can imagine, things where you're not going to have like great repair, like alpha damage is going to be predominant. So M and G2 phase has strong alpha and almost no beta contribution. And then in S phase, where it's the least sensitive, that's where the beta contribution is more significant and the alpha is less significant because you can repair alpha damage. So then there's stuff about chromosomal aberrations, like what things can happen to chromosomes and how they get messed up. I think we'll go into this a little bit more, but breaks occur in single strand of chromatin replicated in S phase. Then later on, when that cell gets to M phase is where you see the damage. In order for the break to occur before the S phase, the cell must be irradiated in G1 phase. So this is kind of just saying like stuff happens in order, and then as the cell gets later on is when things begin to manifest. And we'll get into translocations, dicentrics, and rings later on, but dicentrics and rings are lethal. So that's a chromosomal aberration. So, happening to the chromosomes while they're nice and together. Chromatid aberrations happen while the chromosomes are like dividing into chromatids. So radiation causes break in both chromatids before M phase, seen in M phase. And the radiation must happen after the S phase, but before the M phase. So anaphase bridge is an example of this, and we'll show what that is. The consequences of unrepaired DNA double-strand breaks. You may have heard there's deterministic effects and stochastic effects. It's really easy to get these things confused. I tried to put it straight here. Deterministic effects are organ effects, graded severity with early and late effects, worse with higher dose, and there is a threshold dose. So, like I said, organ effects, cataracts, erythema, epilation, sterility. Stochastic effects are heritable, single cell effects. They're black and white, either present or absent. And it's not like a gradient of severity. So like a secondary cancer, for instance, is an example of a stochastic effect. There's no threshold dose, but the probability is higher with increasing dose. So this is kind of tricky because both of these, like stuff gets worse with higher dose, but in deterministic, it's like the severity gets worse, and in stochastic, it's the probability gets worse. Okay. Any questions about this section? This is a short section, so I'll move on to next. Thumbs up. Cool. So DNA damage and repair is the next section. It wants you to be able to know stuff about molecular mechanisms of DNA damage and molecular mechanisms of DNA repair. We will get into some pathways in this section, which is kind of memorization, but starting with DNA damage. So I think this is something we didn't really learn about, but there are assays to try to measure DNA damage. There's something called a comet assay, where the cells are lysed and then they electrophorese a nuclei, and the fragmented DNA moves away from the nucleus. And so on the thing, it kind of like forms a smear, and the smear is like a comet tail. So it's like fragmented DNA is forming that tail. That's why it's called a comet assay. In order for this thing to work, you have to have a dose of at least 100 centigrade on your sample, or 1 gray. And then there are two things that you can do. One is alkaline lysis. That detects single strand breaks and double strand breaks. This is, I guess, more sensitive. And then neutral lysis, which only detects double strand breaks. So you could think of it like double strand breaks are going to be more obvious. Single strand breaks are kind of more subtle. So if you want to detect single strand breaks, you'd use alkaline lysis. And probably explanation of what these are exactly will pop up in the, but now you know that those are things. This one actually a lot. This is called the gamma H2AX test. It's immunofluorescence microscopy or flow cytometry using an antibody to this thing called gamma protein. This helps detect DNA double strand break damage. So if you're ever curious about DNA double strand break damage, think of this alpha 2AX. It becomes phosphorylated after ionizing radiation. It's very, very sensitive. So I think an example might be like someone got radiated. What's a way you could like, well, someone thinks they got irradiated. What's a way you could look to see if they indeed get radiation or not? And so then you could like get a sample and then do this test because it's so sensitive to radiation that something should light up. And then these are some other assays which were unlikely. There was a section about single lethal hits, accumulated damage, and multiple damage sites. I didn't come across it, so that section is blank. Next is DNA repair. So you can think of it as single strand breaks are easy to repair using the opposite strand as a template. And then double strand breaks are involve either homologous recombination or non-homologous end joining. So starting with single strand breaks, there's base excision repair. This is the most important in radiation. It repairs small lesions. It doesn't include DNA helix distortions. And players involved with base excision repair, there's glycosylase, which removes a base. There's APE1, which is an endonuclease. And then there's either short patch repair, which is for this stuff. There's long patch repair, which involves these proteins. So mostly just like recognition, like which of these things goes with long patch or short patch. Sorry, it's memorization. Another form of single strand break repair is using poly-ADP ribose polymerase, or PARP. Remember PARP inhibitors? So this is like where it comes in. So if you have a BRCA1 mutation, which is important for double strand breaks, and then you use a PARP inhibitor, since PARP is important for repairing single strand breaks, now you're in trouble because you can't do either. Um, and then again, there's short patch repair or long patch repair, which is similar to this. Here's a picture of the base excision repair, showing the glycosylase, the APE1, the XRCC1, and the ligase 3. And you can see how the short patch, includes these proteins, as we talked about, and the long patch includes these ones. So these guys end in the same way, but the way they start is different. This one starts with PARP. Okay. If you ever want me to like freeze so you can do like a screenshot, like just let me know. Or if you want me to go back. There's nucleotide excision repair. This is for bulky helix distorting nucleotide damage. So remember, base excision repair is not good for this. So nucleotide excision repair is good for this. This is almost exclusively for UV light damage, which forms pyrimidine dimers of cytosine, and pyrimidines are cytosine, thymine, and uracil, and 6,4 photoproducts. It involves helicase followed by a ligase. The recognition and repair are separate, and it involves these proteins. There are a lot of rad proteins, but I'm sorry, it just involves a good way to remember is the rad 26 and below. So like all of these numbers are 26 or lower. And then there's this helicase called this that opens DNA strands for nucleoside excision repair.
So we actually
Great.
have to memorize every intermediate protein in these cycles.
Um, Unclear. So you won't beat on all of them, but it's just unclear which ones you will beat on. So um honestly, like I didn't get a lot that were like, oh, what's rad 4 versus rad 13 versus rad something. So the stuff that's bolded uh is more like the higher yield stuff. Other things are like maybe helpful to recognize, but any of the like pure memorization stuff, like I really think that the flashcards were pretty good for. Yeah. Um, and then you can see, sort of just studying this and then doing the radonc questions. If you're feeling like you're doing pretty well, like that's great. Like, yeah. Um, okay. And then lastly, mismatch repair, uh, which involves the MSH proteins. Um, this requires ATP. Next, we're going to get into conditions for altered nucleotide excision repair. These are medical conditions with altered nucleotide excision repair. So there's xeroderma pigmentosum or XP. So they can't repair UV light damage. And then there's conditions with mismatch repair, hereditary non-polyposis colorectal cancer or Lynch syndrome. And I think we've seen this in clinic. Next, double strand break repair. So you can see this cartoon here about kind of when either homologous recombination is occurring or non-homologous end joining. Non-homologous end joining is repairing without the sister chromatid, whereas once you've done the S phase, you can repair using a sister chromatid. Here's a schema of non-homologous end joining. So you have Q70, Q80, MRN, Rad50, DNA PKCS, Artemis, XRCC4, LIG4, and NHEJ1. Um, this is a lot of memorizing proteins, but it's helpful to know what sequence things are happening in. These are just some notes I made about those proteins. Um, and uh, so, okay, and then this is homologous recombination repair. So this one also has MRN, but this one goes with Rad51 and RPA, a DNA polymerase helicase, and some of this stuff. I was a little confused because they both have MRN. And when I looked it up, I found that they don't really know what MRN does in non-homologous end joining, but they just know it's important. Okay, now I'll actually just go to the text here. So for double strand break repair, this is very important in radiation. The MRN complex includes these three proteins, MRE11, RAD50, and NBS1. It senses double strand breaks. MRE is a nuclease, and then MRN attracts ATM to repair double strand breaks, and RAD50 proteins are involved. ATM phosphorylates itself and then others, including these H2A. Remember, H2A came up earlier in that assay, very, very sensitive to double strand. This rapidly localizes to any sites where there was DNA breaks and then serves as a dock for other repair proteins to come in. There are some substrates for these proteins. I don't think I'll read them, but you can see them here. And then for non-homologous end joining, this is now talking about this Q7080 part right here. So this is a dimer that just loves the end of DNA. It binds to double strand breaks quickly and then recruits its friends, which in turn phosphorylates Artemis and other things. There's classic non-homologous end joining, which is direct joining of ends, and that can occur during these phases, G0, G1, or early S. And this is error prone. Often there will be small mistakes. And then there's alternative non-homologous end joining, also known as microhomology mediated end joining. And this is like a crappy backup. It's even more error prone. So you don't want to be relying on this. And then this is just a side note that non-homologous end joining is involved in VDJ recombination, which is important for antibody and T cell receptor diversity. It's kind of a blast to the past. Finally, for the homologous recombination repair, so this is going to be more accurate because you have a sister chromatid helping you. MRN to ATM to CTIP. Well, basically, this is what happens. It's kind of just recognizing more proteins. This is where BRCA1 and BRCA2 are involved, though. So that's kind of cool. And this is like really high fidelity double strand break repair. So hopefully putting some of these proteins in context. And then there wasn't any information to, molecular mechanisms of DNA double strand break, damage recognition, and damage signaling to initiate repair. So I think I just talked about medical conditions where repair is messed up. So there's ataxia telangiectasia. This is an ATM defect. So you could like skim up and see, oh, where's ATM? Oh, ATM's like right here. Um, and then you can understand why it causes problems. It's associated with leukemia and lymphoma. There's something called AT-like disease, and this is due to an MRE defect. Remember, this is part of the MRN complex, and leads to deficiency that's similar to ATM mutation. There's Seckle syndrome, which is an ATR mutation, Niemann-Gem breakage syndrome, NBS, so NBS deficiency, Fanconi anemia, Warner syndrome, Rothman-Thompson syndrome, Bloom syndrome. These may have only come up in like one question at a time during studying, but I just included them for completeness. This was kind of hard to memorize, but I would recognize like ataxia telangiectasia, and I would know that there's such a thing as AT-like disease. And then some of these other ones you can decide how gung-ho you want to memorize them. I put what diseases they were associated with just because I found that to be interesting, but I don't think that ended up. And then remember, since non-homologous end joining is important for antibody and T cell development, if you don't have good non-homologous end joining, you'll have no BDJ recombination, so you'll have severe immunodeficiency. So these are two conditions. DNA histones are important. So if you don't have them, then you're more susceptible to DNA damage. So that was DNA repair and conditions with defective DNA repair. Next is cellular recovery. So I think that this is a little more clinical, maybe a little more interesting. So repair at the cellular level takes 2 to 4 hours, and repair in tissues takes 6 to 8 hours. And here it just shows kind of the timing of different repair mechanisms. So non-homologous end joining is like awesome, it's accurate, and it's fast. Okay, sublethal damage repair. This is repaired on the scale of hours. And if there's a second dose that's given before repair, that can cause lethal damage. They know this from split dose experiments. Dose rate effects and repair. So when we give radiation, often we're giving it at around a rate of 1 gray per minute with a linear accelerator. And that's actually considered a fast dose rate. If we were like in a lab and we wanted to take our time, we could deliver a dose like very, very slowly, like 0.01 gray per minute. And it'd take us like 2 hours to deliver 1 gray. So in this range, there is an effect as you increase the dose, where a higher dose rate is more cell death at that same dose. Okay. This is just remembering the conversion from grays to rads. Once you're above a dose rate of 1 gray per minute, there's little added cell death. Now, this was kind of written before we started studying flash radiation. So flash radiation is like way, 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 way faster. And we're understanding now the effects of that. But this is kind of what the old school rad bio is. Okay. The dose rate effect varies depending on the cell type. Here is a summary. There are flashcards for these, so you don't have to memorize it now. But if you give like 1 gray of X-rays, it causes over 1,000 base damages, 1,000 single strand breaks, 30 to 40 double strand breaks, and then some cross links. Chromosome and chromatid damage. So I alluded to chromosomal aberrations earlier. We use FISH to detect these. And these are good for looking at chromosome translocations. One thing that came up a lot is like above what dose is this sensitive in lymphocytes? So at a low dose of 25 centigrays is when you can start seeing chromosome aberrations. Okay. Um, and then just a small note that if someone had a translocation that was present before radiation, you wouldn't be able to tell if that translocation is due to radiation or not. Dose response relationships for chromosome and chromatid damage. For low LET radiation, like photons, there's a linear quadratic function for how often you'll see rings and dicentric chromosomes because it's 2 hit breaks. Whereas for high LET radiation, it's just a linear function of dose. The induction of radiation-induced terminal deletions, so that's where you like hit the end of it and then it's deleted after the damage, is a linear function of dose. I mentioned this was a section on the outline, use of peripheral blood lymphocytes and in vivo dosimetry. So as I mentioned, lymphocytes can be a good way, if there's damage in lymphocytes, that's a good way to tell if the person got 25 centigrade or more of radiation. Stable and unstable chromatid and chromosome aberrations. So chromatid type aberrations, as I mentioned, occur in S or G2 phase, whereas chromosome type aberrations result or breaks in G1 phase. These are anaphase bridges, which happen from damage in S or G2 phase. Looks like a bridge. And so that's a type of aberration. These are what dicentric chromosome looks like. So this is another kind of damage. So you have a chromosome here. Radiation hits it and splits it up. And then the chromosome accidentally gets confused. It like stacks up each other to form this wonky thing. And then DNA replication occurs. And so you get 2 of them next to each other. This is called a dicentric chromosome because it has 2 centers. This is a ring chromosome aberration. So if you have a damage up top and damage down low, and then this part connects to itself, it'll form a ring like this. And it's important to note that chromosome translocations, these are stable, so they're not lethal, whereas these are lethal. Inversions are also stable. So those are the things that can happen to chromosomes after damage. This was a bigger section. Any questions or should we continue chugging on? And is this pace okay? I'm sorry that there are so many proteins. Maybe John Liu's having fun.
Yeah, I mean, I study a number of these in the lab, so it's all good.
Awesome. Okay, so how do cells respond to radiation? So most solid tumors die via mitotic catastrophe. This is when basically mitosis gets messed up and it leads to apoptosis and the cell dies. This is a little confusing because, I mean, I guess technically apoptosis is a way of death, but if you ever have mitotic catastrophe for radiation, I think you should choose it. Primary apoptotic response is rare and limited to specific tumor types. So lymphoma is unique in that it doesn't undergo mitotic catastrophe and it'll just go straight to apoptosis. Just a side note. What is apoptosis? So we have these proteins from med school, P53, FUMA, BCL2, BID, BAX, caspases, cytochrome C, APAF1, BCL2. Remember, there's the intrinsic pathway, which is in this grainy picture. Okay, so procaspase 8. Okay, this is the intrinsic pathway on this side. And then that eventually leads to procaspase 9, caspase 9, and then these caspases 3, 6, 7 lead to apoptosis. The extrinsic pathway, this is where it initiates based on messages at the cell membrane, and that begins with caspase 8. Caspase 10 is also extrinsic. And these last ones that I said, 3, 6, 7, these are executioner caspases, lead to certain death. And then there was one question that was getting really nitpicky about like what's actually happening. So the plasma membrane is actually intact until the very final stages of apoptosis. And then that's when the membrane blebs off and pinches off to form an apoptotic body. This apoptosis can trigger repopulation in neighboring cells. So caspase 3 serves as a paracrine signal from dying cells to stimulate the proliferation of surviving cells. There is necroptosis. This is programmed cell death, and it depends on RIP3 kinase. So if there are conditions that weren't great enough to trigger apoptosis, there's this other pathway which can lead to necroptosis. And this is programmed cell death that looks like necrosis. And then there's autophagy, which is where cells like eat themselves. And this involves a protein called bacillin. Blah. Okay. Mitotic linked cell death and chromosome aberrations. We talked about a lot of that. Only thing I could add here was, again, DNA damage in G1 and early S leads to chromosome aberrations, whereas stuff in late S, G2, and M leads to chromatid aberrations. Cell division, post-radiation, and time to clonogen death. Cells that are irradiated in G1 could take a long time, staying in G1 for a long time, until finally it leads to death. It could be days, weeks, or even months, depending on the cell type. Also, in G1, they can get chromosome aberrations, which are lethal, but not immediately lethal. So cells can keep dividing for a little before actually dying. On the other hand, cells that are radiated in G2 or M phase are likely to die immediately. Cell and tissue survival assays. Fun. So Assays were pretty intimidating to me because I don't remember learning anything about that in our rad bio lectures. Um, they came up in some of these question banks, but some of the question banks are pretty old. I don't think the assays are like tested that much in the actual test. So the test ended up being like not as bad as I thought it was going to be. But preparing for it was a pain. So there's something called the DNA ladder formation assay. Um, um, I can't really explain it. There's an Annexin 5 labeling assay. Um, so this can detect things during early stages of apoptosis. And then there's this dappy blue dye that looks at the nuclear morphology. I don't even remember ever using this knowledge.
These are all music to my ears, then. No. So the things we actually use in like the year 2023 is tunnel staining, Annexin 5, and DAPI. This ladder formation I've never even heard of. So it seems
Okay.
pretty old school.
Okay. Oh, nice. These are relevant. These happen in real life.
Yes, they do.
Awesome. Um, these are some tissue assays. There's a jejunal crypt stem cell assay. So um, tissue is irradiated in situ, and then response is measured in situ. Um, oh man, I would just like take a picture of this because I don't think I'm gonna go over it very well. Um, there's a skin clone assay, which again is irradiated in situ and response is measured in situ. And there's a kidney stem cell assay, radiated in situ and response measured in situ. Um, there's a bone marrow assay, response determined by transfer to recipient. So this one's a little different, I think. You radiate the host and you ablate their bone marrow. Then you irradiate the experimental subject, the donor, and you do that at different doses. Then you transplant the donor bone marrow cells into the empty host bone marrow, and you're looking for colonies that grow in the spleen, looking for demonstration of pluripotency. Um, if someone was in trouble and needed to restore their bone marrow, let's say from an accidental radiation total body exposure, you can give a gram or a granulocyte colony stimulating factor or GM-CSF. That's all I will say about the assays. Okay, now we get into some formulas, which would have been great to learn about. So you can do a Wikipedia search about Poisson statistics. It's kind of interesting. Um, it's used to describe statistics of small numbers of random events. So something where like maybe 0, 1, or 2 instances happen. Um, and in the case of modeling the average number of hits per cell, we can actually use this statistics. So that's how we get this formula where the surviving fraction is e to the negative d over d0, where d0 is a constant defined as the dose it gives on average 1 hit per target. If there's 1 hit per cell on average, then statistically some cells receive more than 1 hit Some receive exactly 1 hit, and some receive 0 hits. So once you have a dose that you're delivering, D over D0 is the average number of hits per cell. There are 2 models I'll show here. I'll also put this in the chat, I guess. So there's a single hit, single target. This is the simplest kind of kinetics. I think what Walaschak says is if you ever see a graph that looks like this, choose single hit, single target. That never came up, but I was ready for it. This is the simplest model. It happens in M phase with high LET or in cells that lack repair. Okay, kind of makes sense. There's no repair, just stuff dies as you give more dose. This is not an accurate model to explain mammals. Okay, so moving on to more realistic models, there's the multi-target and the linear quadratic models. We are most familiar with the LQ model. And it says in order to be killed, the cell must accumulate enough hits in a short amount of time so that repair isn't capable of repairing all the damage. However, after the first target is hit, the cell might have enough time to repair the damage before the next target is hit. So there's like some probability here. There are 2 dose regimes. There's a low dose regime where you're causing mostly sublethal damage. And then there's a high dose where it's causing lethal damage. And this explains the shoulder. It really bugged me, like no one really said what a shoulder is, but this is what a shoulder is. So if you have like before things start to go down fast, that means it was pretty good at doing sublethal damage repair, like for this whole range of doses. And then conversely, if you had a really short shoulder before like it just started doing awful, that meant its capacity for sublethal damage repair was pretty limited, only in this range of doses. I think I'll get more into that, unfortunately, later. DQ, this is a kind of confusing thing. You can see it right here, but this section, this is like a distance, and so that represents the amount of sublethal damage the cell can get before lethality occurs. I kind of like the way that I just explained it more, that like, you know, for this range, it's pretty good, and then it starts to get bad. So I guess that's what you can think of as DQ. It's kind of like that range until things start to get bad. And then this formula, natural log n equals dq over d0. N is something called the extrapolation number, the number of targets in the cell required to be damaged before losing clonogenicity. Okay. This takes a little bit of looking at and then seeing how it applies in an actual question to digest. So I think I will just leave that there for now.
So for these rad bio equations, are we actually doing calculations or are we just trying to understand the concept? Or
you have access to an online calculator, which is kind of bad and hard to use. I think at most there might be 1 or 2 where you have to like to some sort of logarithmic formula. Um, so I think for the practice questions, you should like be familiar with how to try to apply this, but it might not be a big part. It might not even come up at all. Understanding alpha beta ratio, that's really important. I think I'll just teach this in case they never covered it in the rad bio lectures yet. But this initial slope is what they call like the alpha component of cell killing. And if it's starting to get more death than that, you know that any extra death is now from adding the beta component of killing. So they say that when the projected amount of death that's due to alpha is the same, exactly the same as the amount of death that comes from the beta component of killing. So now these two are exactly the same. That's what we define as the alpha beta ratio. And then we just see what dose does it say here. And that is what the alpha beta is for that tissue. So something with a really high alpha beta means that the alpha component went pretty far before the beta component was able to become significant. So something with a high alpha beta means like the beta component wasn't that important for cell death compared to the alpha. So like skin cells. Okay. You can see how the alpha beta, how the survival curves are different for like low LET, like X-rays, or high LET, like neutrons or protons or carbon ions. Calculations. So this is where there are actually some examples of like plugging things in and like using the formulas. So again, this was the formula. These were what the different things are. And then the example question was, the survival curve is described by dq equals 3 gray and d0 equals 1 gray. What is the surviving fraction at 2 gray for the cell line? And then you had these options. There was a way to intuit this. So I understand this now, but at the time I did not understand it. But remember we said that dq is that portion for like while things are still like pretty good and like repairs happening. So I guess since the dose given is below dq, the cell's still all good. So that's why the answer was 1. This like, 100% surviving. If you did math, we would have used this ln equals dq over d0 and would have gotten natural log n equals 3, n equals 20, and then surviving fraction is d. But this was a trick question. So, but the learning point here is that you can only use this formula when the dose that you're testing is greater than dq. Okay. So let's see if there's another. I think this was just me trying to figure things out. Like this was totally confusing and new to me. So I was like, what is going on here? I think it's handy to know that like e to the -1 is 0.37. That could be useful like on tests or like if you need to do quick math. And then here I was just testing out different scenarios where, okay, I wanted to see what do things look like when the surviving fraction is 10%. So I just did this, and it's like, okay, the dose that you need to give to get something down to 10% is 2.3 times the D0. And again, the D0 is the mean lethal dose, the dose required to produce an average of one lethal lesion. So this is a way where you can figure out what dose you need to give to achieve a certain survival, if you know what the D0 is. Okay, here's another example. Our tumor has 10 to the 10th cells. You want to give enough dose to give the tumor a 1% chance of surviving. 1% chance that there is one cell left. Therefore, you want to achieve a surviving fraction of 10 to the negative 12th. If you know that for S equals 10%, that's 2.3 times D0, then I'm just going to multiply that by 12 because I'm doing like 0.1 raised to the 12th. And so then I get this dose of 27.6 gray. So this is a real kind of question that at least comes up in practice questions. So yeah, it comes down to this formula, and then feel free to take a screenshot of this if you want to kind of see some of the applications. Okay. So a neat thing all of this is that if you have a dose that kills X% of cells, and you also have a dose that kills Y% of cells, then if you add those two doses, you get the dose that kills X% times Y%. Does that make sense? So if 2 gray killed 50% and 3 gray killed 70%, then 2 plus 3 is 5 gray, and 5 gray kills 0.5 times 0.25, since you want to do the surviving fraction. Yeah. Okay. Here's one more example. A tumor has 2 times 10 to the 9th cells. You give radiation that, on average in the population, achieves survival of 10 to the negative 9th. What is the chance of local tumor control? So we have an expected value, which is basically how many cells we started with, what the average survival is. And so the average expected survival is 2 cells. But since we're given a like what's the chance of having 0 cells surviving, because that's what tumor control is, we've gotten it down to 0. Here we're going to use the Poisson distribution, this probability mass function formula, which I showed it up here, but I showed it in an example down here. So I'd plug that in and I would get this answer. There was a 13.5% chance that all the people have 0 cells surviving and they all get cured. More simply, when estimating the chance for cure, k equals 0, the formula will always be p equals this, where m is how many cells you're starting out with, and s is a surviving fraction. Okay, we're almost done with this mathy section. Hang in there. Let's see, for acute doses with the linear quadratic model, this one we're most familiar with. S equals e to the negative alpha times the dose minus beta times the dose squared. For low dose rates, beta approaches 0. So you can just cancel this out and you get a survival of just e to the negative alpha times the dose. So here are two examples. Let's estimate surviving fraction. You can take pictures of these if you want, but you can see how it works out. Okay, these are some homemade graphs I made because it was hard to understand them with the existing materials. So I explained this is what the shoulder is, right? It's the dose something can handle before the survival starts to drop a lot. Late responding tissue has a lower alpha beta, so it dips down. It's more like sensitive to high doses at a time, whereas early responding tissue has a graph that's more like just a pure alpha component, and then maybe towards the end starts to dip down like a beta. So early responding tissue is less curve shaped. Late responding tissue is more curve shaped. Fractionated dose. So when you have one, if you were just giving all the dose at one time, your curve might look like this. But if you, in this example, we're just going to give dose 2 grays at a time. So we get this first part, then the second part, then this third part, etc. So this is me illustrating that. Interestingly, the LQ model is showing what does it look like if we have infinitesimally small doses at a time. So the LQ model is actually something plotted with like, 0.1 gray, then 0.1 gray, 0.1 gray, 0.1 gray. That's how you get the curve. So I thought that was interesting. Fractionation makes much less difference for early responding tissue. And then the survival curve resulting from fractionated protocol is referred to as the effective survival curve. Okay. And then there's something in the textbook called iso effect curves, which is based on a bunch of experiments where they looked at different types of tissues and they got kind of the same effect in those tissues along these lines. So if you gave like less dose per fraction, but like some higher total dose, that was similar to giving high dose per fraction, but less total dose. So this graph is actually kind of backwards if you look at the x-axis. Okay. Late responding tissue shown in red. So remember, skin has some component of early response and some component of late response. Um, Late responding is low alpha beta, so it has steep iso effect curves. Early responding has high alpha beta and has shallow curves. Okay. I think that's everything. This was a big section for sure. This next section is actually, I think, kind of more fun. It just has to do with LET and oxygen. So, the definition of LET, energy transferred per linear unit distance along a particle's path. We've said this before, the difference between low LET and high LET. There's something called chromothripsis, which is shattering of the chromosomes. So high LET can cause that. RBE, relative biologic effectiveness. How effective the radiation is at cell killing. It's the ratio of doses required by two radiations to cause the same level of effect. So this is super confusing. This is actually worth like wrapping your head around, at least so you get a bunch of questions right on the practice questions. So an example of RBE applied is like I have a dose with 250 kV photons, and I want to know the ratio over a dose with a different type of radiation. So it could be MV photons, or it could be MV protons, or uh, lower keV electrons. So the numerator is always some dose with 250 kV photons. And the denominator depends on what the question is. So I guess if I was doing something with like, I don't know, carbon ions, I probably don't need very much dose of carbon ions relative to the dose I would need with kV photons to achieve the same effect. So I would say my carbon ions have a high RBE. Okay. If I had something that was weaker than kV photons, then the dose that I give with that weak type of radiation is going to have to be more than the dose I give with the kV photons. So I'll have an RBE that's less than 1. Okay. Okay. So let's go into some examples here. Gamma rays and X rays. So if I use X rays with 250 kV, my RBE is 1 because I'm comparing apples to apples. 250 kV to 250 kV. If I'm using slightly weaker, so sparsely ionizing photons, so when I have like 1 MV to 15 MV, I actually have an RBE that's less than 1, slightly. If I use protons, protons are more damaging than kV photons for the same dose. So protons have an RBE that's greater than 1. Neutrons have an RBE that's 2 to 8. And then iron, argon, and other heavy ions have an RBE that's high, but less than 8. We've talked about this in lecture, but there is a limit where you increase the LET, you get higher and higher RBE, but then you get to a certain point where it becomes counterproductive. I think you're literally just like blasting past like so fast that you're missing out on being able to give some damage. So that's why we have this less than 8, because it like peaks. And then if we have higher LET than 100, we might actually start to get lower RBE. Okay. The other thing in this graph is the oxygen enhancement ratio, which I'll talk about lower down. As RBE increases, the oxygen enhancement ratio decreases. That means heavy particles rely less on oxygen enhancement to cause damage. Whereas if you have weak photons, they really need oxygen to cause damage, and they'll have a higher OER. Does that make sense? Okay. They want you to know about the effect on RBE on change in fractionation. RBE increases as dose decreases. This is weird, but think about it. If I gave 1,000 gray with with anything, I'm probably going to cause like a similar amount of death. But if I gave just like 1 gray, that difference between what I'm achieving with 250 keV photons is going to be much more pronounced. 1 gray of like carbon ions is going to do a lot more. It's going to exaggerate that difference. That's key concept 1. Key concept 2. As you increase the number of fractions, you are increasing the RBE. So take a look at this Ben-Lee custom graph. We have single fraction shown in black and fractionated shown in red. If it's solid, it's neutrons with high LET. And then dashed is like our comparator, the 250 keV X-rays. So the RBE is going to be kind of like the ratio of these. Sorry, the ratio of the doses. Okay, to cause the same amount of death. So here I'd have this versus this. Whereas if I do fractionated, I have this versus this. So this looks like if I fractionate, I have a higher RBE. When fractionated, high LET particles versus 250 keV X-rays have greater relative kill at a low dose compared to high LET particles versus 250 keV with single fraction. So this one takes a little bit of staring at, but if you get this, I think you'll be good. You'll like understand what RBE is, and the questions will be much less confusing. Oxygen enhancement ratio. This is also another pretty specific definition. Like it seems kind of intuitive, like, oh yeah, it's like how much oxygen matters. But specifically, it's the ratio of radiation doses that produces the same surviving fractions under 2 different oxygen conditions. It's been measured in experiments. OER of 3 is what we typically say for low LET particles. OER of 1 for high LET particles. oxygen enhances radiosensitivity. The enhancement plateaus as you increase oxygen. So you're not going to get much more kill by going from like 98% oxygen to 99% oxygen. But you will get a bigger difference when you're going from like a small percentage of oxygen to a medium percentage of oxygen. Cells reach full radiosensitization at 20 to 40 millimeters of mercury. And this is near the oxygen concentration of venous blood. And then you can think about like stuff in other places, like in thin air. Yeah. So these are some bullet points, which are, I think, the gist of what you need to know. You might have a question that's like, between which range does the OER change the most? And it'll say like from like 0.5 to 1%, or from like 11% to 12%, or from 80% to 82%. So you would choose the one where it's a low percent and the graph is really steep. This is, I think, useful clinical information. How far can oxygen actually travel from an artery? So it can travel 150 micrometers. That's at the arterial end. And then the venous end, where you don't have as much, it travels a little bit less than 70. Therefore, if you have a tumor with a radius that is small enough that like wherever there's an artery, it should get the whole tumor, that tumor will not have a necrotic center because oxygen's able to get to all of it. But once tumors get bigger and they start to like outstrip that arterial supply, that's when you start to have necrotic centers. And what is 200 micrometers? That's 0.4 millimeters. So we've been like looking at CT scans, we see like giant masses and the inside's dark, and we're like, oh, that has a necrotic center. Yes, but even small tumors, anything that's bigger than 0.4 millimeters, also has areas where there's little areas of necrosis. So this is like the model that we think about, but this is actually a misrepresentation of biology. And it actually looks more like this, where you have a tumor, and then within the tumor, there's lots of little areas of necrotic centers. And so when we're talking about like oxygenation in tumors, it's not just like, oh, is the center of this giant mass getting oxygen? There's also like parts all up throughout the tumor where the oxygen effect is at play. Okay. Hypoxia can be either chronic or acute. Chronic is due to limited diffusion distance, whereas acute is due to acute changes like vasoconstriction. There can be reoxygenation when an acutely hypoxic cell becomes oxygenated. And there can also be slow reoxygenation of chronic hypoxic tissues over time. OER is greatest during S phase and lowest during G2 phase. HIF alpha, so this was like related to the Nobel Prize. Under hypoxic conditions, HIF1 alpha is active and promotes angiogenesis. Unoxygenated conditions, on the other hand, HIF alpha is hydroxylated, and VHL ubiquinates hydroxylated HIF1 alpha during normoxic conditions to send it to the trash. Gets ubiquinated. The enhancement effect from oxygen is more pronounced at high doses. So in the high dose range, OER is 3.5, and low dose range, OER is 2.5. And remember, we said most of the time we think about an OER average of about 3. So two things with OER. One is like what kind are you using high LET or low LET? And how does that affect OER? And the other thing is like how much dose are you given? And that also affects your OER. OER versus LET. I kind of mentioned these. Impact of oxygen concentration. We talked about the plateau. We talked about this. This was random trivia. Humanidazole detects hypoxic cells. Mechanisms of oxygen effect. So yeah, this is kind of cool. You like finally learn like what's actually happening. Because I think we've heard like several different explanations over the years of like, oh, it's like, hydroxyl radicals, or oh, but it's actually this, then that, or this, then that, then that. But the full thing is shown here. And kind of has time frame too. So ionizing radiation hits water molecule, becomes ionized, produce water ion radical, which is charged and has an unpaired electron. Very unstable. This then reacts with another water molecule to form hydronium ion and a hydroxyl radical with a short lifetime. And then the hydroxyl radical can transfer their free electrons to organic molecules like DNA to form organic radicals. Organic radicals have a slightly longer lifetime before being reduced by oxygen to an organic peroxide, which finally causes irreversible damage. And the whole thing happens in milliseconds. You won't need to know anything more detailed than that. You might not need to know very much of this, or you might. Okay. How are you guys doing? Is this more than what you were expecting? Do you want me to go faster? We can get through the end. I think we can do it. Is it okay if I get to some and I just say like, take a picture of this. This is a lot of memorization, and then just focus on the concepts.
Yeah, I think so.
Okay. I don't expect you guys to memorize any of this right now, by the way. It's just so you can like have seen the words and heard them once.
Yeah, and I think like the concepts that weren't really well, just
Okay.
like alpha beta and stuff.
Okay. Okay. So far as the concept explaining, has that been okay-ish?
Yep.
Okay. Okay. Okay, great. Tumor assays and TME. So now we're um I might skip. These are just definitions. Like what's a xenograft? What's a tumor regrowth delay assay? What's a tumor regression assay? What's a local tumor control assay? Um, I think I'll focus on this because this is the most important assay for curative effects of radiation. It's the dose that achieves 50% tumor control. You radiate the tumor in situ, and then you also evaluate the response in situ. Um, Actually, all of these are in situ. I guess it's helpful to know because you might be wondering like, oh, so this is a different excision assay. This is you radiate the tumor in situ, and then you take it and put it on a dish, and you evaluate it in vitro. So that's why I was like including this in situ here. This, although it's like a gold standard, it's very labor intensive and costly. And also it doesn't control for the immune response of the host. So it's like an imperfect measurement. It's not just showing what the effect of radiation is. Whereas if you do an excision assay and you like take it out of the host, now you've like gotten rid of the immune system. So it gives you like a nice pure look. But now the downside is that you're the response is not being measured in the original environment, so you don't know if it's actually accurate. So I guess there's pros and cons of these different things. Um, and yeah, this is what the graph looks like for like a tumor control assay. Again, follows a Poisson distribution. Experiments that study the impact of a radiosensitizer around the TCD50 dose will be most sensitive for detecting an impact. I think this kind of makes sense. Like, if you want to know, is my thing having a difference, like my new drug, look at it somewhere where the graph is steep. So if it's helping it, you should get a sharp spike in the signal. Okay, great. How does a tumor grow? It's exponential at first, but then so V stands for volume. So initial volume times e to the a times t, where a is just some constant that you determined, t is time. But then as its size grows, the growth fraction decreases and cell loss increases. So late in its life, its growth has this divided by b component. This is called Gompertzian growth. Okay, tumor microenvironment. This is someone's favorite topic. So TME is a vasculature, extracellular matrix, fibroblasts, and the immune cells that get in it. Radiation can affect the TME. There can be targeted effects and non-targeted effects. And TGF beta is one thing that we can track in the TME that mediates DNA damage repair. TGF beta suppresses anti-tumor immunity, hence protecting cancer cells. So one potential way that we could optimize the killing from radiation is to reduce TGF beta. How does the TME regulate tumor growth and vasculature? Myeloid cells promote regrowth by enabling a tumor stroma that promotes growth. Angiogenesis and neovasculogenesis. Tumors have lots of things that promote angiogenesis. How does it respond to hypoxia? There's the HIF1 alpha pathway, and then some other stuff, and then VEGF, and then vasculogenesis, and then tumor regrowth. Clinical consequence and relevance of hypoxia in tumors and tumor progressions. We've talked about oxygen is required for indirect radiation damages, and hypoxia affects radiation response and recovery. Reoxygenation after radiation. Some people have described tumor response in 4 stages. The first, the endothelium is affected. Then there's tumor cell death. Then there's early regrowth of vessels, and then late tumor recurrence. I don't really know if this like came in, but it was described. I don't know if this is a universal model, but it's interesting. Ways to non-invasively detect hypoxic regions in tumors. So these are different imaging techniques. Listed here. Cellular and molecular responses to hypoxia and hypoxia-induced signal transduction. Mentioned the HIF-1 alpha pathway, seems like it's important. And then cellular composition of tumors. So cancer cells commonly use glycolysis for energy, even under normal oxygen conditions. So when they're doing aerobic glycolysis, this is known as the Warburg effect. Therefore, lactate is not a surrogate for tumor hypoxia. It's going to be producing lactate even when there's normoxic conditions. Immune microenvironment and the role of inflammation. If you ever do some sort of like immunology course, it'll talk about there's elimination, then there's equilibrium, and then there's tumor escape. And this is the natural cycle of immunity and cancer. The role of inflammation. So we have the innate immunity and the adaptive immunity. The innate immunity is not cell specific, it's immediate, just bombards things. Neutrophils, basophils, eosinophils, these are granulocytes. And then there's mast cells and there's macrophages. There's M1 macrophages, which kills enemies and stimulates the immune system. And there's M2 macrophages, which release IL-10 and TGF-beta, poor cancer prognosis. You have dendritic cells, also as part of innate immunity, and natural killer cells. So interesting, natural killer cells are part of innate immunity, but they help trigger the adaptive response. So now we have the adaptive immunity, which is cell-specific. It's developed by antigen-presenting cells, dendrocytes that migrate to lymphoid tissue. So we talked about NK cells, T cells with T cell receptors. There's helper T cells, the CD4 plus, binds to MHC class 2 molecules. And then you have the T killer cells, which are CD8 plus, and they bind to MHC class 1. I always think it's like 4 times 2 or 8 times 1. This is some extra granularity about the T helper cells, so like TH. So a naive T cell can become a TH1, TH2, TH17, or a T regulatory cell. And TH1s stimulate cell-mediated response to get more T killer cells. TH2 inhibits cell-mediated response and promotes a humoral response. T regulatory cells suppress the immune system, and TH17 can become either TH1 or Treg. So helper cells can have diverse functions. And then lastly, part of the adaptive immunity, we have B cells. They have B cell receptors, which respond to antigens. Antigens are presented by T cells, or antigens that are presented by T cells are T cell dependent antigens. So these are foreign proteins that the T cells got from the dendritic cells, give it to the B cells so they can form anti-cancer antibodies. And then antigens that are independent of T cells are not important for cancer. And the abscopal effect. So the definition is out of field disease control. So it's not saying like specifically what's happening. It's just saying that, hey, we're noticing something out of field happening. As stated here. There's one buzzword here, STING, stimulator of interferon genes. And this pathway can help evoke the abscopal effect via type 1 interferon production after radiation. So sting abscopal effect. Cancer biology. Exciting. Wow. Okay. Here's one more formula. This is called cell loss factor. It ranges from 0 to 1, and it's how many cells are lost as the tumor grows. So if the cell loss factor was 0, that means the tumor would just keep on growing and growing as you predict it to grow. But we know that as a tumor gets bigger, it's going to start to outgrow some of its stuff. And so it's going to be growing and dying at the same time. So this is just a formula to describe that. If the cell loss factor is 0.6, that means for every new cell produced, 0.6 cells die. So the cell will grow at a rate of your baseline rate times 1 minus 0.6, or 0.4. Growth fraction, the fraction of cells that are actually growing in the tumor. Cell cycle time, the time for cells to divide into 2. So you can do a formula to see what's the potential doubling time, the teapot. You assume that your cell loss factor is 0, and then you have your teapot equals TC, your time for your cell cycle, divided by the growth fraction, how many cells are growing. And there's this gamma correction factor. So I guess this is how my formula is a little bit corrected. You might have it says like the cell cycle time is 4 minutes and the growth fraction is 80%. What's the potential doubling time? And you have to be like, ooh, I know these things go together somehow. And then you can figure it out. Propidium iodine staining is a dye that binds DNA and stains cells. It tells you what cell cycle cells are in. If the cell is in G1 phase, it will have 2N DNA. If it's in G2 or M phase, it has 4N DNA. And if it's in S phase, it has something in between 2N and 4N. 3-H-thymidine and bromodeoxyuridine labeling is used to estimate the time length of cell cycles. It has an antibody incorporated into the S phase. Okay, I don't think you need to know the specifics, but these things are used for length of cell cycle. Here's some rules of thumb for the cell cycle. G phase is very variable in length, or G1 phase is very variable in length. Earlier I was saying it could be like, hours, days, weeks, months, depending on the cell. S phase typically is 8 hours. G2 phase typically 4 hours, and M phase about 1 hour. This is a summary of sort of what's happening during those different phases. Okay, this is a key chart. It's totally just memorization, but I just read this as like DEAB, like DEAB, and I just remembered that D starts with like the beginning of the cell cycle, like G1 to S. Then there's E, then there's A, which is S to G2, and then there's B, is G2 to M. It's my representation of this graph here. So it's saying what proteins are involved in cell cycle control and initiating checkpoints. So cyclin D goes with CDK6 and 4. Cyclin E goes with CDK2. A goes with CDK2. B goes with CDK1. So it kind of goes like 6, 4, 2, 2, 1. And that's the main thing. Like this, what I've highlighted here is like the main thing you need to know. This wasn't really like who are the promoters. It looks like it's kind of the same across it, except E2F is in this column. And then who are the regulators of these different transitions? I've summarized that here. But just remember DEAB and then 642211. That this comes up a lot. It's this is a key graph. Okay, great. Promoting the cell cycle. Okay, I won't go over this. I basically explained it above. Here's one thing to note. CDK1 is not the same as cyclin D1. I think I was confused for probably like half of my studying, so you don't have to make the same mistake. Here's some checkpoints. Great. Phases of cell cycle and radiation sensitivity. Oh, cool. We talked about some of this, where homologous recombination and non-homologous end joining. How homologous recombination is better and nicer, but you can only do it once you're in the S and G2. And now non-homologous end joining, it's like not that good. Okay. Molecular signaling. So I think Harish would be really excited here. Like all his alley. I like the emoji, Lisa. Okay, so we have different signaling pathways and proteins involved. They're listed here. These are all different kinds of kinases: ATM, ATR, CHEK1, CHEK2, PI3K, MAPK, ADM, ATR, mTOR, and DNA PK are all phosphatidylinositol-3 kinase-like kinases, or PI3K. Okay. Phosphatases. So there's P10. Here, oh, not yet. Okay. So molecular signaling pathways activated by ionizing radiation. So we radiate, what happens? Radiation causes inflammation and increases TNF alpha. P53 and NFKB are activated at even very low doses. If you touch a cell, these pathways will become activated. AP1 is another transcription factor that leads to inflammation. EGR1 is a transcription factor that leads to growth factor expression. These did not come up. NF kappa B pathway. I think highlights are that NF kappa B activates TNF alpha, IL-1, and IL-6, as well as some of these other things. Transcription factors. So there's a P53 pathway. ATM phosphorylates P53 to activate it, and when it's active, it regulates the cell cycle. There's these E2F transcription factors. These will promote cell division. This is pro-tumor. And cell death pathways. We talked about the intrinsic apoptosis and the extrinsic apoptosis. I've just listed the proteins involved for each of them. And then BCL2 is an anti-apoptotic. These are actually my initials, Ben C. Lee. So I protect against apoptosis. Okay. I think this was I think this may have been the screenshot from Harisha's presentation. But a lot of human cancers have a mutation in the tyrosine receptor kinase or RAS signaling pathways. And then these are some drugs that target different parts of it. And there is a section in the RadBio about drugs and what pathways do they target. So this is kind of helpful to know. There's a really great spreadsheet actually that's in the Res Drive, which has a summary of like how to remember the names of certain drugs. I think I just like skimmed that like the 2 days before for like very short-term memory stuff. Okay. You should know like which ones are BRAF inhibitors and which ones are ALK inhibitors. Mechanisms of cancer development. Okay, this is kind of conceptual, but I feel like it's kind of intuitive, so I'm gonna skip over that. Common oncogenes. Okay, so oncogenes promote cancer. Um, these include these guys. I list them one at a time here. This is a bit dense, so I think I'm not going to go through it, but you'll need to know like what are the oncogenes, what are their pathways, what kinds of cancers do they tend to be found in. I've bolded key things. And then transcription. Okay. This, I feel like if you were a lymphoma oncologist, you would know these, but just be familiar with some of the really commonly tested translocations, like 922 for CML. Other ones might be common, but I honestly don't remember them, so I don't remember. Um, okay, there can be oncogene deletions. So NRAS deletion in neuroblastoma, also BLIM deletion in neuroblastoma. I listed this one, but I don't remember using it. And there can be amplifications. So NMYC amplification, also neuroblastoma. LMYC amplification in lung cancer. HER2 new, also known as ERBB2 amplification in breast cancer. We see that one. And then KRAS amplification in lung, ovarian, and bladder cancer. So I think it's neat to see how these are all oncogenes, and they've been affected either by a translocation, deletion, or an amplification. Then there's tumor suppressor genes. So we have a lot of our guardians in DNA repair, like BRCA1 and BRCA2. These are tumor suppressor genes. Some are known as gatekeepers. These are survival checkpoints, and then some are caretakers, which are like helpful for repair. Gatekeepers also send cells to apoptosis or senescence if things don't get fixed. There's this MDM2 ubiquitin ligase that normally keeps p53 levels down, but when DNA damage occurs, p53 becomes phosphorylated, and then MDM can't stick to it. So then p53 is able to do stuff, and so MDM2 is kind of like the lid to p53, but the lid comes off when p53 is phosphorylated. P53 activates a lot of things, or it's a transcription factor for a lot of things. And then p53 can be inactivated by point mutations and certain proteins. So HPV has an E6 protein that blocks p53 activity. Adenovirus can block p53 activity, and SV40. If there's too much MDM2 activity, too much lid on p53, that can also inactivate it. And then if you have loss of INK4A ARF, this leads to too much MDM2 activity, which then leads to p53 inhibition. Okay, we talked about E2F up here. Somewhere up here. And retinoblastoma inhibits E2F, like so. So this will stop the promotion of entry to S phase of the cell cycle. So yeah, understanding that pathway. There's the P16 pathway, protein 16, also known as CDKN2A. This slows progression from G1 to S by inhibiting CDK4 and CDK6. Remember the table thing up here, CDK4 and CDK6, G1 to S. And this prevents phosphorylation of the retinoblastoma protein. RB stays active and bound to ETF and stops promotion of the cell cycle. P16 is inactive in most pancreatic cancers and some melanoma, and it's overexpressed in HPV, head and neck squamous cell carcinoma. The mechanism in HPV squamous cell is that there's a viral protein, HPV E7, which leads to degradation of RB, which leads to increased E2F and uncontrolled cell growth. The decrease in RB releases P16 from its negative feedback control and results in increased P16. So you see high P16, but it's actually not P16 that's causing all the problems. It's actually this protein. So P16 is like the signal that something was messed up. There's the P10, another tumor suppressor, NF1, Wilms tumor protein, VHL, this, that, and then NF2 for Merlin. There's a section on telomeres. So you should know what a telomerase is and how it relates to TERT, which is a catalytic subunit of telomerase. And TERT promoter is often mutated in glioblastoma. So if you have hyperactive TERT, you have more telomerase, and then you have unlimited proliferation. Signaling abnormalities. Cells normally want to fix themselves or die after DNA damage. Anything that interferes with this response is abnormal. Cancer is a genetic disease. Yeah, there can be problems with DNA replication. There can be epigenetics, such as Barrett's esophagus, and epigenetic reprogramming, like changing how stuff grows, epigenetic silencing, methylation, and then this is also implicated in aging. And then when epigenetic changes from the parent aren't inherited, it's called imprinting. Multi-step nature of carcinogenesis. I think we sort of know this, but you have only one out of many cells eventually makes it to cancers. There's a selection process, clonal competition, Olympics. Then there's mutation and genetic drift, and then instability creates possibility for cancer. 30 doublings is equal to about 10 to the ninth cell, which is equal to 1 gram or 1 cubic centimeter. We talked about T potential and T volume. You can estimate the volume of tumor as 4/3 pi r cubed. For small tumors, the T volume should be similar to the T potential. This is the time for doubling volume, but for large tumors, it takes a lot longer for the tumor to actually double in size because there's lots of cells dying and sloughing as the tumor grows. Carcinomas have a high cell loss factor. Sarcomas have a low cell loss factor. And we see sarcomas can get really, really, really big. Interesting. I actually just made that connection now. I'm like learning as I'm going. Okay. Significance of tumor characteristics. Large tumors have lots of cells, so there's a higher likelihood of resistant cells existing after chemotherapy. This is why chemotherapy can't cure bulky disease, because all you need is one resistant cell to make a new population of resistant clones. So chemotherapy is better at eradicating microscopic disease. Yeah, sometimes treatment can drive further cancer evolution. That's bad. Cancer stem cells, these can self-renew and also give rise to daughter cells. They could be more radio-resistant and chemo-resistant due to things, MDR1 expression, high antioxidant levels, slow cell cycles of reprogramming. Cancer genetics and genomics. Modification of histones that surrounds the DNA can lead to signaling that directs the packing and unpacking of the DNA double helix. This can be regulated with acetylation, phosphorylation, methylation, or ubiquitination. Nitrosylation does not occur. That sounds like a multiple choice question. Main epigenetic alterations. I had nothing to contribute here and nothing to contribute here either. And I passed. Omic approaches to cancer. Comparative genomic hybridization. You're looking across the entire genome, and you can compare the cancer's genome to a normal cell's genome. So that's what comparative genomic hybridization is. There's Sanger sequencing. Now it's usually just used to identify mutations in a single gene. Next-gen sequencing, or NGS. Shotgun sequencing. This is also, okay, fragments of long strands of DNA are randomly generated, and then those fragments are sequenced, either Sanger sequencing or next-gen sequencing, and then they're pieced together like a puzzle. And ultimately, they make one single continuous DNA sequence for the complete length of the chromosome. Circulating tumor DNA. Biomarkers in cancer. I literally just tried to go by this. So PCR-able Philadelphia chromosome. And then the only thing about that is Gleevec or imatinib targets that. Molecular profiling of cancer. Oh, I had no idea what chip was, so I put this video here which explains it. I will put this in the chat. It was a nice explanation. Great. So you might have something about electrophoresis. Like, I think you will. So it's definitely important to know like what's a Western blot, what's a Northern blot, a Southern blot. So Western blot detects antibodies to proteins. And this is the section on proteins, right? Then we have this section down here on RNA. So northern blots detect DNA, RNA. Southern blots detect DNA, DNA. And so there's this acronym, which is really nice, called SNOWDROP. Southern, Northern, Western, DNA, RNA, protein. That was pretty helpful. Genomic alterations. What did you need to know about this? Single-stranded conformation polymorphisms. This is electrophoresis separation of single-stranded nucleic acids, which results in a different secondary structure and measurable differences in mobility through a gel. This was rather dry. I don't remember this coming up. I think it was in an old practice exam, so I wrote about it. But these are ways you could study single nucleotide polymorphisms. And this was the biggest section. The rest of it becomes more clinical. We're doing okay. Like, there's only 3 more after this one, and these ones are shorter.
Everyone else. It's not a lot.
Yeah, do you think we could maybe take a break for today?
I'm cool with that.
Well, I don't know what everyone else thinks.
I'd be down for that.
Like,
Getting
My brain is going to explode.
Okay, good. I mean, my brain is
Seriously,
gonna
this is so
explode too.
helpful though, Ben. Oh,
Really?
man.
much more than what we've had all year.
Yeah.
Yeah, you guys
Yeah, I'm
got
just like, I
this.
can't absorb anything more.
Yeah, this was great. Like this um yeah, this was the biggest section, and then the rest of this, I think, coming in with like fresh minds, you'll be like, it won't seem that daunting. Yeah.
Like,
Um, yeah. Cool guys.
I don't know if you want to kind of figure out when.
Do we plan on tomorrow at 8 then?
Sure.
That sounds okay to me. Oh boy. Yes, tomorrow at 8.
Maybe there will be a little bit of some stuff